So Tony, Steven, Jin, and I have tyrosine kinase receptors. First off, we're going to start with what a receptor is. Receptors are protein molecules, which are usually found embedded in the plasma membrane surface of the cell, and these allow the cells to receive chemical signals from outside of the cell. I will be drawing a diagram, but this shows a receptor detecting hormone. Once received, the cell will elicit a biological response from it. Now I'm going to open this in Notability. No, excuse me. All right. Let me get my cells. So this is going to be a cell. These are the receptors. And this is going to be maybe a hormone, because our receptor is going to receive those. Now, Cameron, would you like to tell me what where the hormone would go for the signal to be received inside the cell?
To the receptors.
Correct. Great job.
The binding site.
All right. Here's just some facts about them. Tyrosine kinase receptors are in a family of receptors with similar structures. They are transmembrane proteins that act as receptors for growth factors, neurotrophic factors, and some other extracellular signaling molecules. They make up, or tyrosine kinase receptors make up over half of the tyrosine kinase genes. I believe there's also like inhibitors, there's a few other ones I think. And most of these tyrosine kinase receptors are like single subunit receptors. There's a couple exceptions. Alright.
Tyrosine kinase receptor is a receptor that receives signals that regulate growth of the cell. And tyrosine is a type of amino acid that's on the tyrosine kinase, and kinase is an enzyme that catalyzes the transfer of the phosphate group. And this is a process. There are several parts to the tyrosine kinase. And there are signal binding site, which is these little things, and tyrosine on polypeptide. These are the tyrosines. Those are amino acids, and alpha helix that's embedded in the membrane. This part. And this is step one. The signal molecule will attach to the thing on the binding site, and this will cause the two monomers to bind together to become one big molecule, and it's called this will form a dimer, and this is called an phosphorylated dimer, and it's also called activated tyrosine regions, and they're ready to receive phosphate. And step two is tyrosine kinase takes a phosphate group and of an ATP, which leaves an ADP and inorganic phosphate. The phosphate will then attach to a tyrosine, and this will turn the unphosphorylated dimer into a phosphorylated dimer. And it's fully activated receptor tyrosine kinase.
Alright, so here we have a phosphorylated dimer, and in our example we only have 6 activated regions, but it can have up to 10 or more. And these activated regions are going to be the sites for the relay proteins in the cell to bind and thus create a cellular response in the cell. So the relay proteins, once bound, they create the cellular response, and they send, these cellular responses can vary between, an example, mitosis or meiosis, which is the separation of the cell, creating two cells, basically growth, or it could send other responses. And it has 10 or more responses from one single tyrosine kinase receptor. So it's a very precise system, and one ligand can activate many multiple responses in the cell, and they either hasten the growth of the cell or slow it down, which is very important for the cell because uncontrolled growth would be very, very bad, and we'll get on that later, Jim. So why would this be important for humans? As very large multicellular beings, why would this growth factor be very important for humans? Albert.
Because we have a lot of cells, so we need
Right,
a lot of cells.
there's a lot of growth that goes on in a human. For example, in the stages between infancy and childhood, there's a lot of growth that goes on with the human. And it'd be very important for this to slow down, otherwise it would just be a big mass of cells and we'd just never stop and we'd die. So it's very important, well, it's the truth. It's very important that this system is regulated, and this regulation occurs within the cellular responses that occur from the relay proteins. So onto the mutations.
So like, when tyrosine kinase cannot regulate it, like it became like mutation, and it existed in the nature, like naturally it happens. So like, the position of tyrosine kinase receptor is like permanent on, so which means that it works without any control. So it grows like unregulated like growth. So this is like known as cancel, and this is right. So this mutation like does not require the signal molecules to activate it. So this mutation causes that tyrosine kinase to form like phosphate continuously, and the phosphate attached to the relay proteins to like, and it cannot like regulate the growth of the cell, so it keeps growth like continuously growth. So here's the concept that no more tyrosine kinase picture, but if you see the mutated tyrosine kinase, it will look like this, that the relay molecules would not be necessary to like activate this tyrosine kinase